So, hello, I'm Florent PERRIN from Carotenoid team for quality and resistance of vegetable, and I will speak about the regulation of carotenogenesis and carotenoids accumulation in carrot roots. So let's begin by a very good question: why study carotenoids? They are for plants natural secondary metabolites which are accumulated in different tissues, and due to their antioxidant properties, they are very important for photoprotection. As you can see on this experiment, an Arabidopsis mutant which overexpresses a gene from carotenoid biosynthesis pathway is more resistant to intense solar stress compared to the wild type. Moreover, they are attractive pigments in flowers and fruits for pollinators and disseminators. Carotenoids are not only important for plants, but also for us. And because we are not able to synthesize these pigments, we need to take them in our food. And due to their provitamin A activity, they are important to prevent diseases. As you can see on this photograph, there is 4 golden red generations which have been created for Asian people. Moreover, due to their organic properties, they are important for food and vegetable quality. The carotenoid biosynthesis pathway is relatively well described, and all genes and metabolites have been characterized. The geranyl pyrophosphate is the precursor of this pathway, and is important because it's the precursor of other major metabolites in plants like cytokinin, gibberellin, and chlorophyll. In carrot roots, there are 4 main carotenoids which are accumulated: lycopene, alpha-carotene, lutein, and beta-carotene. And the catabolism of these carotenoids lead to the synthesis of abscisic acid and strigolactones that are important hormones for plant defense. So the aim of this presentation is to do an overview of the regulation of carotenogenesis and carotenoids accumulation in carrot roots at different scales. We will see the genetic diversity, transcriptional regulation, enzyme activity, cellular compartmentalization, and environmental impact. So, what about genetic diversity? These works show you that depending on color type, there are 1 or 2 main carotenoids accumulated. Red genotype accumulates mostly lycopene, orange genotype alpha and beta carotene, and white genotype accumulates no carotenoids. Moreover, it has been shown that depending on color type, total carotenoids contents are different. For example, in orange type, there are more carotenoids compared to the yellow types. In addition, results which are not shown on this presentation describe that in the same color type, there are different contents of each carotenoid. And this diversity is due to an anthropic selection which has been focused on color roots. And this map shows you the different domestication centers of carrots. We can see that in the Middle East, purple and yellow types are mostly represented. In Europe, there are orange and white type. And in Asia, pink and red types. To go further, researchers wanted to know how carotenoid trait is transmitted during successive generations. And it has been shown that there are 2 major Q-tails in the lineage group 2 and 5, which are correlated with carotenoids accumulation in carrot roots. In addition, 2 loci have been identified. The first one correlated with dilem color, and the second one with yellow trait. More research works have shown that carotenogenesis genes are correlated with carotenoid accumulation, and for example, nucleotide polymorphism of XEP is correlated with total carotenoid contents in carrot roots. But till now, there is no correlation between Q-tail, loci, and carotenogenesis genes. So, what about genetic diversity? We have seen that there is a large diversity due to an anthropic selection, and genetic markers have been identified and correlated with carotenoids accumulation. But a more accurate mapping will allow to determine if QTL, loci, and genes are correlated between them. And this work could lead to control color trait heritability in carrots for crop improvement. Now, what about gene expression? These results, obtained by Jérémy Cloteau, show the gene expression during the development of 4 carotte genotypes: a white, a yellow, an orange, and a red genotype. And we can see that for the red genotype, for example, lycopene content is correlated with gene expression during the development. However, we can see that for the white genotype, which accumulate no carotenoid, transcripts are present. So, gene expression partially explains carotenoids accumulation in carrot roots. And one of the most striking results of this work is that Psi gene seems to be correlated or responsible for carotenoids accumulation in carrot roots. Moreover, it has been shown in tomato fruits that gene expression and especially Psi expression is correlated with lycopene accumulation during ripening. But to validate if Psi gene is important for carotenoids accumulation, a functional validation is necessary. And Mas and collaborators have transformed a right carotte genotype to overexpress Psi gene. And the results show that the mutant present an orange fruit phenotype and high carotenoid amounts. So, these results provide evidence that Psi is an important key for carotenoid accumulation in carrot roots. Moreover, it has already been shown that in potato tuber, the overexpression of Psi increases lutein and beta-carotene accumulation in tuber. So, what about gene expression? We have seen that gene expression seems to partially explain carotenoids accumulation, but in ripening fruits, gene expression and metabolites accumulation work as an on-off switch, while in carrot roots, gene expression and metabolites accumulation evolve gradually. Moreover, we have seen that Psi gene is an important key for carotenoids accumulation in carrot roots. But this gene expression is genotype dependent and does not allow to establish a general trend. And it will be very interesting to study the catabolic gene expression to determine another important regulation step for carotenoids accumulation. Now, what about tissue organization? Baranska and collaborators have shown by Raman spectroscopy that there is an heterogeneous distribution of beta-carotene among the root section. And beta-carotene is mostly accumulated in phloem compared to xylem. At a thinner scale, chromoplast structure have been investigated by microscopy by Kim and collaborators, and they have shown that in orange type, as you can see here, chromoplast contains more crystal forms of carotene and low amount of starch. However, in white type, chromoplast contains more starch and looks like an amyloplast structure, as you can see here. In addition, it has been shown that the origin overexpression, the origin is involved in the differentiation of proplastid to chromoplast. The overexpression of the gene in cauliflower and potato tuber leads to an high accumulation of beta-carotene. So we have seen that there is an organized distribution of carotene among the root section, and chromoplast organization and differentiation seems to be important for carotenoid accumulation. Moreover, it will be very interesting to study molecular regulation of chromoplast differentiation in carrot roots. Now, what about impact of environmental factors? Recent works have shown that water supply impact carotenoids accumulation in carrot roots. Embody and collaborators have worked on sprinkler irrigation during 2 years, and Zeipina and collaborators has worked on sprinkler and drip irrigation during 2 years. And these results are conflicting and depend on the year, but we can see that water supply impacts carotenoids accumulation. Moreover, Allegria and collaborators have worked on storage conditions, and they showed that different storage treatment impact carotenoids accumulation during carrot storage. So we have seen that irrigation system and storage treatment are important for carotenoids accumulation in carrot roots, and also for carrot quality. It will be important to study more environmental factors that could modulate root quality. And a very important question that remains is what the impact of environment on different regulation scales seen previously. So, in conclusion, I've completed the carotenogenesis scheme with the different regulation scales. We have seen that genetic control, gene expression, and especially Psi expression, chromoplast differentiation, and environmental factors are important for carotenoids accumulation in carrot roots. But it will be important to study the organ specificity of different gene isomers, for example, or epigenetic regulation, post-transcriptional regulation as splicing or degradation, and translational regulation as protein maturation or protein degradation. Thank you.
You've shown that irrigation and water stress was involved in modulating the expression of Psi. Have you looked at some impact on abscisic acid as well, in this case, and some interaction between the two?
In carrot roots, there are no works on that, but it has been shown that, for example, the overexpression of ZEP involve an accumulation of abscisic acid and leads to resistance for Arabidopsis mutant. But in carrot, there are no study on that.
I'm working myself on abscisic acid and the impact it has on diabetes in humans. And maybe it could be an idea of eventually, since we're working a lot in this pathway, to have a high abscisic acid carrots, which could have an impact on metabolic diseases.